This is a review of the acid-base balance lab. An acid is a chemical that can dissociate to release hydrogen ion. A strong acid is one in which most or all of the molecules of acid will dissociate when mixed in water. The pH becomes very low when a strong acid is mixed in water. An example is hydrochloric acid with a pH of about 1. A weak acid is one in which some, but not most, of the molecules of acid dissociate when mixed in water. The pH is somewhat low, and the lower it is, the stronger the acid. For example, lactic acid has a pH of about 2.5 in water, making it weaker than hydrochloric acid, but stronger than carbonic acid, which has a pH of about 4.5. A base is a chemical that can form a bond with hydrogen ion. In this way, it removes free hydrogen ion from solution. A strong base is one in which most or all of the molecules will pick up hydrogen ion. A good example of this is hydroxide, which will combine with hydrogen ion to form water. A weak base is one in which some, but not most, of the molecules will pick up hydrogen ion. An example is bicarbonate ion. A buffer is a combination of a weak acid and its own base. For example, carbonic acid becomes bicarbonate, and bicarbonate can become carbonic acid. The pH scale is a measure of the concentration of hydrogen ion in solution. The way the pH scale works means that adding acid lowers the pH of solution. There are 3 important chemical buffers in the blood. These include carbonic acid bicarbonate, phosphate, and proteins. Carbonic acid and bicarbonate are diagrammed here. Carbonic acid, H2CO3, can dissociate to release hydrogen ion, forming bicarbonate, HCO3-. Likewise, bicarbonate can pick up a hydrogen ion to form carbonic acid. So the 2 chemicals are linked by the addition or removal of a hydrogen ion. Phosphate buffer works basically the same way. H2PO4- can give up a hydrogen ion to form HPO4-2. HPO4 can pick up a hydrogen to form H2PO4. Proteins are abundant in the plasma of the blood and in the cells of the body, and many of these proteins carry negative charges that can reversibly bind with hydrogen ions, so proteins are important buffers intracellularly and in the blood. A pH indicator is a chemical that changes color with different pH levels. In this lab, you used phenol red as a pH indicator. As you can see in the image, the higher the pH, the redder the phenol red will be. And as you add acid, the color changes from red to orange to yellow the more acid is added. In this lab, you blew through a straw into a beaker with water colored with phenol red. You might wonder why carbon dioxide can be an acid. The answer is that carbon dioxide will spontaneously react with water to form carbonic acid. Carbonic acid is classified as a volatile acid because it can be blown off as CO2 when you exhale. So blowing into water with phenol red will turn the solution first orange and then yellow. Bicarbonate is a base which elevates pH, but also a buffer to added carbonic acid. So adding bicarbonate and phenol red will make your solution take longer to turn yellow while blowing into it, as the bicarbonate picks up the hydrogen ion formed when carbonic acid dissociates. Buffers prevent pH changes. In humans, the normal blood pH range is 7.35 to 7.45. Anything below 7.35 is called physiological acidosis, even though it's still in the alkaline range of the pH scale, since it's above 7. It's considered acidosis because it's below the normal level for human blood. Physiological alkalosis is anything above 7.45. That's blood pH. Other body fluids will have different normal pHs. For example, the normal pH of stomach secretions, because hydrochloric acid is secreted when food enters the stomach, is about 1.8, very acidic. The normal pH of urine can range very widely from an acid pH of 4.5 when the body is in acidosis and needs to excrete acid, up to a high of about 8 when the body is in alkalosis and needs to excrete base. The normal value of human urine is around 6, since we usually eat and produce more acids than bases. An acid-base imbalance occurs when the pH gets outside of the normal range. Acidosis is a blood pH lower than 7.35, and alkalosis is a blood pH higher than 7.45. Acidosis can be caused by a respiratory imbalance or a metabolic problem. In a respiratory imbalance, if you have too much carbonic acid, you're retaining too much, that's going to be because you were hypoventilating. Hypoventilatory disorders can include things like asthma, COPD, or simply suppression of the respiratory center in the brain. Metabolic causes can vary from too much acid being swallowed, too much acid being produced, or even renal disease. Ketoacidosis is a condition we see in people with uncontrolled diabetes mellitus. The acid comes from metabolic problems in the cells. But other causes can be as simple as diarrhea, which causes a loss of excess bicarbonate from the body. Alkalosis can be respiratory or metabolic as well. Respiratory alkalosis occurs when there's not enough carbonic acid, and that is due to hyperventilation. Typically, hyperventilation is associated with anxiety or panic disorders. Metabolic alkalosis usually occurs when either too much alkaline is ingested or too much acid is lost. Consuming too much of an antacid can cause metabolic alkalosis, as can vomiting, which causes a loss of stomach acid. Compensatory mechanisms are those changes in the body that are designed to keep the pH in the normal range. Chemical buffers are our first line of defense against acid-base imbalance. They will act to counter pH imbalances immediately, but you only have a certain amount of buffering ability in the blood, and you cannot compensate for large or continuous imbalances. Respiratory compensation can match a metabolic imbalance, so you can hypoventilate to hold on to carbonic acid to balance a metabolic alkalosis, or hyperventilate to excrete carbonic acid to balance a metabolic acidosis. However, you can't hypo or hyperventilate forever. Renal compensation can control bicarbonate excretion, and this can be quite long-term, but usually slower acting than respiratory compensation. Your kidneys can increase hydrogen ion excretion to correct for acidosis, thereby keeping bicarbonate in the blood to buffer excess acid, or increase bicarbonate excretion to correct for alkalosis, and this will cause a drop in blood bicarbonate. Here we see our two opposing conditions. In the left diagram, we see an increased blood hydrogen concentration, which means a decreased pH. Here we see some various causes. Typically, excess ketoacids and loss of bicarbonate are going to give you a metabolic acidosis. You're going to balance that by having the kidneys excrete excess hydrogen ion. In the opposite condition, the pH is too high from too low of a blood hydrogen concentration. This is going to be an alkalosis. This could be from excess antacids being ingested or loss of hydrogen from vomiting stomach acid. In this case, the kidneys are going to balance that by excreting the excess bicarbonate. Changes in respiratory rate likewise can cause acid-base imbalance. An abnormal increase in the respiratory rate means more CO2 expired, which means less CO2 in the blood, and a decreased CO2 in the blood means a lower amount of carbonic acid and that is going to elevate the blood pH. If you decrease the respiratory rate, that's hypoventilation. Less CO2 expired means more CO2 in the blood. More CO2 in the blood drives the increase in carbonic acid production, which will decrease the pH. So hypoventilation causes acidosis. Hyperventilation causes alkalosis. Here you see our three buffer systems at work. Chemical buffering through phosphate, protein, and bicarbonate buffering systems can work within seconds. The respiratory compensation, either increasing or decreasing respiratory rate as a balancing mechanism to offset a pH imbalance in the blood, can work within minutes. Renal mechanisms are slower. It takes hours or even days for the kidneys to fully compensate for either a metabolic or a respiratory disorder. In this lab, you're also required to analyze arterial blood gases. We identify the acid-base imbalance by evaluating the arterial blood gas values. Step one, of course, is to know what the normal values are. pH, normal, 7.35 to 7.45. Carbon dioxide partial pressure, 35 to 45 millimeters mercury. Bicarbonate concentration, 24 to 28 milliequivalents per liter. Step two is to evaluate the patient's ABG values. Is the pH too high, which would be alkalosis, or too low, which would be acidosis? Is the PCO2 too low or too high? Is bicarbonate too high or too low? If either PCO2 or bicarbonate concentration is normal, then there is no compensation. Step three is to correlate the abnormal values. A high CO2 or a low bicarbonate will cause acidosis. A low CO2 or a high bicarbonate will cause alkalosis. Here's a handy tip. If you're looking at some example values and you notice that all three values are abnormal and match, then it's a metabolic disorder with respiratory compensation. If you have a low pH, a low CO2, and a low bicarbonate, then it's a metabolic acidosis. If you have a high pH, a high CO2, and a high bicarbonate, then it's a metabolic alkalosis. So if the values don't match, it's a respiratory disorder. Low CO2 causes high pH, high CO2 causes low pH. This concludes our review of the acid-base balance lab.